His graphics make sense if you have the kind of prescription drug coverage he does. This is the last section of the human biology class. I don't know if I'm going to get it all on one video or not, but it's the last section. This is the way that people tend to see the medical establishment. This might be the most powerful tool yet developed to separate a fool from his money. Doesn't necessarily apply just to medical stuff, but prices for medical treatments are just out of control in a lot of ways. Now, some of it is you get what you pay for. Is that they variety and the number and the power of treatments that are available to Americans is only comparable to some very well-developed countries, and is, in a lot of senses, probably better than theirs. But it involves expensive developmental costs, it involves expensive investment costs for machinery, all these new devices that are coming out, and personnel, lots and lots of personnel, lots and lots of processes. And so it is a very, very expensive endeavor. We don't allocate the money particularly well. It could be cheaper. It could be more organized. It could be a lot of ways that we can do a better job in terms of medical costs. But I'm going to kind of give you an idea of where some of the costs come from. One thing that I refer to a lot of times is that there's a blog written by a fellow who's a pharmaceutical chemist who's at the kind of the ground level of developing new pharmaceuticals. And it's called In the Pipeline, if you want to go look it up. It's written, depending on which entry you read, it's fairly readable. When he gets into the molecular stuff, even I'm looking, I can kind of skim it and get a grasp, but, and the comments are always entertaining when you don't get too much political talk in there. But people are in the sciences and they're doing testing. Testing is very often generated from what's called basic research. This is stuff that tends to come out of university labs, where somebody will go, okay, we've taken a look at this little part of the cell, and this is what it seems to be doing, and this seems to be the proteins or the other chemicals involved. And somebody at a pharmaceutical company will go, oh, that probably relates to this disease. If we could manipulate that chemistry, we might be able to affect this disease, maybe even cure it. So they're always reading the basic research. And you'll hear people go, well, yeah, but why don't the government's funding all of these university programs, then why aren't the drugs being developed for free? Well, because universities are not developing them for the most part. Usually they're just kind of giving the basic idea. And then somebody goes, well, if this is true, then this should happen. And there's two levels there. First, if this is true, and a lot of times this research doesn't pan out. It turns out that this is preliminary. What they thought was happening isn't actually happening. And then the second part is, okay, if this is what's happening, then this is what should happen. Often doesn't work the way you expect it to. Cells are incredibly complex systems. Our bodies are incredibly complex systems. And knowing a little piece of it, what they call reductionism, doesn't necessarily mean that you can go after it and do anything reasonable with it, because other stuff gets in the way. If you're going to test, you need to design a test. Science is a much more creative endeavor than people give it credit for, because you've got this idea that you want to test. Well, how do you set up the test to do it? Well, usually you focus on, this is what I think is happening. Okay, how do you test that? That's always what science says. How do you test it? How do you test it? Sometimes you go, I don't know. It's an interesting idea, but there's no real way to test it. And then it's kind of a dead end. People kind of toss the idea around. They play with it in simulations and such, but it may not ever go anywhere because it isn't really testable. And you can't really dig into it if you can't test it to see if your hypothesis is true. But usually, if you do have a test available, how do you do that? Well, let's see if this is doing what I think it's doing. So let me set up a test to see if it's doing something. Okay, I got a test, I get results. Well, how do I know if the results are actually connected to what I'm testing? Let's set up a matching test, but I'm gonna test this thing, take this thing I'm testing and change it. In a drug test, I can remove it entirely. If I'm giving people injections of my chemical, in my control test, my comparison test, I'll give people injections, but it won't have my chemical in it. Results here versus results there, you can probably go, well, that's because of the chemical working. That's why these results are different from those results. Other times you have to, what if we vary the amount of sodium in somebody's diet? Well, that would be a dangerous thing to completely cut out. So you compare a normal sodium diet to a reduced sodium diet, and then look for differences in, and then you gotta pick a result. What are we gonna measure in order to decide what's actually going on? And that is a tricky thing that happens in the vaccine trials, is that, okay, we're gonna give people a vaccine, we're gonna have a control group that they're also getting a shot, they're getting a placebo. So they think they're being treated. As far as they know, they're being treated the same way. Because our mind is a weird thing. It has lots of effects on things. Usually not whether you get coronavirus or not, but how quickly you recover could be affected by whether you think you've been vaccinated or not, because your immune system does respond to certain mood and other chemical inputs from your brain. So the placebo group, as far as they know, they're getting the treatment. They've signed a paper. They know they could be getting a placebo, but everybody hopes. The people in the blog I talked about, a lot of them have been in the studies and they go, oh, I know I got the placebo because my arm didn't hurt after I got the vaccine shot. And other people going, oh, I got the vaccine. My arm's been bothering me for days. So placebos are tricky. You don't want your two groups to know which group they're in, because you're looking for the effects of everybody think they're being treated, but that's part of your design. So you got your comparison groups. You need lots and lots and lots of people, because in any small group, if you have an oddball in your small group, they're going to skew. You're going to average your group, and your group only has 5 members in it, and one of them reacts in a really odd way, then your whole group average is messed up. If you have a group of 1,500, and you have that oddball in your group of 1,500, he's not going to affect your average very much. So numbers are a big deal. Early on, the coronavirus tests were either being uncontrolled, no comparison group, or done on very small numbers, or done in what's called a retrospective fashion, where they were kind of trying to compare them to populations that weren't really in the tests, and you don't really know how they were treated. So that starts to get very, very tricky. You want as much grab on your test. I want to say control, but control has a specific meaning in tests. You want as much grab on your tests as you can. It's just having people out and about in the real world is already an issue for what's called confounding factors. Other things that creep in that mess with your results, but they're not actually what you're testing. Sometimes those produce what are called artifacts. The placebo effect, the effect on people's health of just their feeling that they were treated, is an artifact of the act of treating them. You get placebo effects from, the more dramatic the treatment, the more dramatic the placebo effect. Acupuncture, bunches of needles stuck in you, has a huge placebo effect. But if you set up a control group that thinks they're getting needles put into them, because you're using fake needles that don't actually go into the skin, they disappear as you push down, and then they stand there. The two effects on whatever health condition you're looking at are virtually identical. So real acupuncture, totally fake acupuncture, produces an effect because you can have control over pain and a lot of things, but it's a placebo effect. It's an artifact of the actual treatment. Oops, that's not a dash. I just happened to put the pen down in the wrong place. This is a very specialized type of study. The pharmaceutical blog that I read, these guys are trained in this. So they can look through the details of a vaccine study and the chosen endpoints and the number of people and how they set up the controls and be able to go, okay, they did this fine, this one's real, this is really iffy, oh, this is totally wrong. Is that I don't know, I don't even know why they released this information except maybe to get more investment money in their company. And their peers, they know how this stuff works, and so they have a good critical eye on it. And good science should be peer reviewed before it gets out to the wide world. But with something like coronavirus, where there's like a clock on everything, there's a lot of stuff released before it's effectively peer reviewed, and then it's kind of peer reviewed on the fly. But of course, it's in the wide world that people who don't understand how this works go, well, what about hydroxychloroquine? But the eventual studies that were large and controlled found virtually no effect of hydroxychloroquine on the eventual. And what are you measuring? Are you measuring symptoms? Are you measuring hospitalizations? Are you measuring having to go on respirators? Are you measuring mortality? It turns out that when you measure all of those, you don't see a huge impact of hydroxychloroquine. But those are the late studies, the early studies, where they had like 8 people, and they're looking more at symptoms, which are more controllable by placebo effect. They got what looked like good results, and very often in a small group. I'll talk about this later. You get good effects with a small group. I'm going to talk specifically about drug development. It, more than any of the other things that you can get, follows a very scientific method approach. Devices, how they're tested, when they're tested, it's not as regulated in terms of the steps and the checks and the reviews and making sure that everything's okay before it goes out there. And in fact, there's an awful lot of physical treatments, both devices and surgical treatments, that people just decided that they worked, and nobody's really done an organized study of them. And, or when they do, they very often go, well, these stents that they follow up arterial blockage, the bubbles. When they put a stent in there to increase blood flow and keep the artery from closing back up, if you look at subsequent heart attacks or mortality as your measuring point, they don't really do anything that it doesn't compare to just not having them in there. But medicine is very trendy, very set in its ways. They like humans in general. They go, well, I believe it works, and yeah, your study said that, but you're not convincing me because I've seen it work. I've never really done a controlled study, or I've done it on everybody, so I've never really seen what happens when you don't do it. But damn it, it works. Drug development, because of the review process, is a little bit more controlled. One thing you need to know about drug development. You put a patent in on your potential drug molecule, whether you got it from academic research or you've got this fancy artificial intelligence computer, which doesn't seem to really do much that people can't do. And it goes, here's a molecule that could be useful in this particular endeavor, in this particular condition. So you send the molecule down to the lawyers, and they put in a patent application. And when the patent application is approved, only you have a right to whatever comes from the development of that particular molecule, unless somebody else has something similar, and then you fight in the courts about it. But you got 16 years from, let's put it in, to, okay, now the patent is lapsed, and anybody who can produce the molecule and sell it can do it, and they don't have to pay us any money. Drug development very often takes 8, 10, 12 years before you can actually market if it gets approved. 90%, over 90% of these molecules are never ever going to be sold. They're going to be tested to one level or another. So there's money going to be invested in them, but they're not going to make it all the way through the pipeline. That's why the blog is called In the Pipeline. All the way through to the end where you can actually start to make a profit on them, which is part of the reason why at the end they come out and they charge humongous amounts of money on them. Right now, the patent system has always been a little iffy. It's been a little bit more broken lately, where a company will take a molecule that it makes and they'll twist it a little bit, or they'll change the formulation of it enough to be able to apply for a new patent on it. Like, it's insulin, but it's, insulin, it's more readily available insulin, or it's slower releasing insulin, or it's something, or this, or it's that. And whereas a lot of the old patent reviewers would go, but it's the same thing. All you've done is play with it a little bit. We're not getting you a new patent on that. That's been happening much more lately. So you'll see a drug come out that's just a little modification of a drug that was out, except they now have a renewed patent on it. And they probably got it through the pipeline much faster because a whole bunch of things about it had already been approved. So, and the other thing too, once your patent has lapsed, generic drugs are produced by other companies that don't do the research. They do need to prove to the Food and Drug Administration that their version of your molecule will be effective. They don't need to hit your levels. They need to hit somewhere north of 70% effectiveness. But they could put it in a pill that works a little bit differently. The drug companies are very careful formulating these things so that they deliver out of your digestive system and through your liver and into your general circulation and into your cells in as good a way as they can do. And they spend money on all of these different mixes of pills, even injections and that sort of thing. The generic companies don't have to match that. They just need to be able to produce the chemical and show that it is effective up to a certain level, but not up to the original level. So sometimes people on generics go, Doc, it's not working as well as the other thing. And the doctor goes, oh, it's the same chemical. Well, it is, but it isn't. And in some cases, what they mixed in reacts with somebody in a way that the original mixture didn't. So some people go, well, I'm on the generics, but it's making me dizzy. And the doctor goes, no, that's not what that chemical does. That's not a side effect. Well, it may have to do with what they call the binding agents. You have a whole handout about all these mixtures that they use for drugs. So generics are not necessarily generics. Here's another thing about generics. A lot of the recent generation of drugs are what they call biologics. Very, very complex molecules that mimic biological molecules. They are very expensive to produce, and when the patent lapses, a lot of the generic companies go, we can't make any money on that. And so the original company just keeps selling it at the prices that they originally set. Nobody's going to come in after the fact and go, here's a cheaper version of that, because they've looked at the molecule and gone, we're going to have to build a whole new factory to make these. It isn't worth it. Let them sell it. We'll stay with the other stuff that we've got on the market. Okay, that's a good place to pause. We'll pick up the actual bits and pieces of drug development in the next one.